let's talk about phylogeny. So we'll be covering molecular phylogenetics here. There are actually different ways of doing this, and they all try to get at the same idea, which is to figure out, of all the things that exist right now, how did we get here? Where's the last common ancestor of each group? And if things have a more recent common ancestor, at least in genetic terms, you would say they're more closely related, basically. So we're trying to hierarchically categorize all of life and reconstruct, essentially the way that life developed. Let's look. So you can do this with characters of any kind. That's what they call it. Now, characters can be physical traits, like the size of a nose. You can make a family tree based on that. But what we're going to do is use characters that are derived from molecular biology. So that's why we call this molecular phylogeny. It's going to be RNA, DNA, protein, basically. And I think protein does the most powerful phylogeny work because there are 20 different amino acids, but there's also conservation at the amino acid level because proteins are generally making some kind of machine, and the machine has to have a particular shape in order to function. It can't just change everything all at once or it will probably break. And if it's broken, it will probably not do its part in keeping the organism that hosts this gene alive. So, in other words, those disappear from nature all by themselves. Okay, some definitions. Read them, think about them. Okay, you got it? Yeah, phylogeny, reconstructing ancestry, essentially. And so, we're going to make trees, which are our little graphic summaries. And these are all basically based on an idea that Darwin happened to articulate in his notebooks. I don't remember if it had been seen elsewhere before that, but the idea makes sense. Just like a family tree with little branch points, except that we end up with species rather than individuals, the way we tend to do it. All right, so these characters, come in different character states. So would say different character states are the different nucleotides you could have. Now, to be the same character, you have to show that there is homology, that these are the same thing. So, for example, I could compare noses of different primates and probably come up with a phylogenetic tree that may even reconstruct something useful. Who knows? But if I were to include in there, say there's some kind of fish that has a nose that doesn't function anything like our nose, then probably that is just going to corrupt the data. So we have to show that these share a common ancestor, that they come from the same developmental pattern, from the same genetic stock, something like that. We have to show that we have homology, basically. And if we can't validate homology, you can't make a tree. The thing about phylogenetic tree-making software is it will always give you an alignment and it will always give you a tree, no matter how bad the data you feed in is. And this is a problem because people will feed in bad data and then get out a tree and say, well, the computer says it branches like this, even though it makes no sense whatsoever. And it was just an artifact of a poor data set, poor data inputs. So there you go. So for it to be the same character, we have to show that every one of these amino acids or nucleotides, at least within a reasonable probability, does indeed match in some way the ones from another organism. And so it's alignments that are going to show this. If you get a significant alignment, if you build these little words or these little groups together and get a nice big long chunk that aligns pretty well to another chunk, you assume the statistics are for the entire chunk, which is nice. So you can say, well, the entire chunk has a great E value or a great P value. And so therefore, we will make the assumption that all of the parts of this region have an equally good P or E value, and so we can use the data. It's an assumption that is the best you can do, but will occasionally be wrong. And through no fault of yours, just mostly from inadequate data. A lot of times if you're working on something that's very different from everything that's been seen before, you will not get as much help from homology, easy to find homology anyway, as you might like. So there you go. All right, character state definitions. Yeah, so the characters can be many different kinds. So we can, let's see, we can have a plesiomorphy, which means that this particular version has the same gene as its ancestor. Now, if we have DNA from, let's say, my ancestors, if you were to dig up great-great-grandma and take a little of her DNA, you could probably find the regions of my genome that are essentially plesiomorphic, that are identical, possibly a little bit rearranged or slightly mutated, but identical with what she had. Yeah. And I guess if it's slightly mutated, it would not be a plesiomorphy at those nucleotides where the mutation is. It would just be a plesiomorphic region in general. Apomorphies are differences. Plesiomorphies are similarities. You're like, cool, yeah. And so apomorphies are what we call derived states. Derived means it's different from the ancestor. So if my great-grandma had brown eyes and I don't, then we would say, oh, I have a derived trait. Yeah. Doesn't matter how it was derived. It just is derived in the sense that it is different from the ancestral condition. And I bet you can spot, one of the flaws in here, which is that nobody ever has just one great-grandparent, right? That's not how math or people or biology works. You got to have at least two, I think. Yeah. Even in a European royal line, you got to have at least two. And so really, this ancestral state should be taken as a pool of people or genes or whatever we're talking about, whatever we're measuring on our chart, which together contributed to the current state. So an ancestor doesn't have to be like one cell or one individual. It basically can't. It has to be the individuals that together gave the DNA, through whatever mechanism that actually led to the present state. There you go. Yeah. Then my favorite are syn apomorphies. So syn means same, S-Y-N. And apomorphies are differences. So they're same differences. It means that all the members of this group that are closely related are going to have this same feature, which is different from the ancestral condition. Synapomorphies are a good way that we use to define species. Now, the other thing I'll say is that all taxonomy is both relative and somewhat arbitrary. The means to obtain taxonomy are very precise, highly mathematical, not at all arbitrary. The arbitrariness is where you decide, I'm going to draw the line for species at this different. And usually those are done on a case-by-case basis in some way that makes sense to one or more people. And that's about all you can say. There's quite a range there. The concept of species, genera, all these things, these are very human classifications. People are good at grouping things together that are similar. We're good at finding, human faces in a piece of bread, stuff like that. We are good at identifying underlying characters that can be used to organize groups of things. And so this is just people doing it with animals. So while what we decide as a species is going to vary depending on which species we're talking about, it may or may not have a whole lot of basis in genetics. Yeah. And many species were named before the advent of genetics. And so when people come back and look at the genes and then say, wow, these two things are not at all related that are in the same species, or these two things in the same genus should not be in the same genus, or these three things that we thought were each a separate genus are actually all almost identical at the nucleotide level. They're just like a different color or something. There's one gene that has a big effect on shape. In those cases, yeah, the species definition can be wrong. So we're not going to worry so much about species per se. We're just trying to reconstruct phylogenies with generic ancestors that go back there. Practically, we're going to end up using some of these generic ancestors as de facto lines saying, well, everything that's related to this group that comes from this node on the tree. A node is where some branches separate. We're going to call those mammals, something like that. Does that make sense? I hope so. Yeah. So synapomorphies, very useful. They define species, for example, or taxa. Yeah, there are synapomorphies that define mammals, for example, lactation, hair, stuff like that. Autapomorphies are things that are different about a particular individual species, whatever it is. These show evidence of change. They can give us evidence of the amount of time that's passed since they shared a common ancestor with something else, because only some parts will be changed. If everything changes, then you can't really say that two things are related because there's no way to prove it unless you have the intermediate steps. And most of the intermediate steps, as far as life goes, happened 1 billion or 2 years ago. Most of the really interesting ones anyway. So yeah, you're not going to recover them from molecular data, most likely. You have to reconstruct them. And so that's what phylogeny is. It is the art of reconstructing an ancestral tree that makes sense and is based on defined characters that theoretically anyone with the proper technology could go out and find and define. And then people can add to a tree that exists or rearrange a tree based on new data. And those are all fine. So with that out of the way, the big thing to talk about is going to be, well, how do I make this tree? And that is a whole story in itself. That's for next section.